A variable is any factor or condition that can be changed in an experiment. Can be soil type, can be rainfall, it could be the breed of the animal, it could be temperature, it could be anything. So anything that is going to change in an experiment is your variable. And we have three types of variables. We have independent variables, dependent variables, and controlled variables. So your independent variable is what you change in the experiment, okay? I for independent, I for I change, okay? That's what I always tell the class. I for independent, I for I change. What is the one thing you are changing in your experiment? So that either they look at their experiment there, more than likely they're going to have two situations there. What is the difference between the two beakers? What is the difference between the two soils? What is the difference in your experiment is your independent variable, okay? So it could be as you were saying, it could be temperature, it could be soil type, it could be type of milk, it could be anything. Okay, so your variable, independent variable, is what you change in the experiment. Your dependent variable is what changes as a result of the independent variable. So I always tell them that your dependent variable is what you measure. Okay, so your independent variable is what you change. Your dependent variable is what you are measuring. Okay, so you could be measuring the soil drainage. You could be measuring the DM of the crop. You could be measuring the organic matter of a soil. You could be looking at the amount of bacteria in a milk sample. Anything like that is whatever you are measuring is going to be your dependent variable. And then lastly, it's your controlled variable is all other variables that are kept the same. So I don't think maybe it's explained enough, but when you're carrying out an experiment, you are only changing one thing at a time. Everything else is kept the same. You have the same graduated cylinders, you have the same volume of water, you have the same type of soil. Everything is kept the same bar the one factor, which could be temperature. Okay, so everything is kept the same. Everything that is kept the same in your experiment is your controlled variables. The one difference between your experiment is your independent variable, and what you are measuring in that experiment, whether it be temperature, time, anything like that, is your dependent variable. It's like anything. If your car isn't working, you're not going to change the gearbox, the clutch, do the timing belt, change tires all at the one go. You're going to do it problem by problem. If the gears are grinding, look at the gearbox. If it's not the gearbox, then move on to the clutch, then move on to something else, okay? You change one thing at a time in every experiment that you're doing. Bit of a graphic there I use. So your independent variable, what you change. Your dependent variable is what you observe or measure, and your control variable is what you keep the same. So in this example here, Mary and Alan want to investigate if water is needed to grow cress seeds. They place 5 cress seeds in a dish labelled A with cotton wool and some water. They place another 5 cress seeds in a dish labelled B with cotton wool and no water. It was later noted that the cress seeds that received the water germinated. Discuss the 3 variables in this experiment. Right, so we just write out here what we have in our bit of a blurb. So we have dish A, we have 5 cress seeds, cotton wool and water, and dish B we have 5 cress seeds, cotton wool and no water. And we were told up here that the cress seeds that were water germinated. Right, so what is our independent variable? So our independent variable is the change. Okay, what is the difference between dish A and dish B? What's the only difference between the two is your independent variable. So your independent variable, hopefully it's as clear as day that you can see that dish A was watered, dish B has no water, therefore that is your independent variable. Then your dependent variable is what we are measuring. Okay, so the dependent variable, what we are measuring in this experiment. In this experiment, we are measuring whether or not the cress seeds germinated. So our dependent variable is plant germination. Okay, you could be measuring the height of them, you could be measuring the length of the roots, the length of the shoots, but in this experiment, we are just told that we are measuring germination. Okay, whether or not the crop germinated. And then our control variable is what stayed the same in our experiment. What is the similarities, or what is in common with dish A and dish B? Dish A, we have 5 cress seeds. Dish A, we have cotton wool and cotton wool. Okay, so they are controlled variables. So number of cress seeds and cotton wool. Another good one to put in is environmental conditions. So you're not putting one in an oven and one in the freezer. So same sunlight, same humidity, same rainfall, same whatever. Okay, so in the same environment. So if you're ever stuck in the exam for a controlled variable, you can always put down environmental conditions. So hopefully that explains the difference between your independent variable, dependent variable, and controlled variable.